From the JAMA Network, this is JAMA Surgery Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Surgery.
Welcome, JAMA Surgery listeners. This is Amalia Cochran, the web and social media editor for JAMA Surgery, coming to you today to discuss intraoperative wound irrigation for the prevention of surgical site infection after laparotomy. My guests today are Dr. Tara Mueller and Dr. Daniel Reim from the Department of Surgery at the Technical University of Munich School of Medicine and Health. Welcome to both of you today.
Thank you very much.
Thanks for the kind invitation.
Listeners, we have two abbreviations you may hear during the course of today's recording. The first of these is SSI, which is surgical site infection, and the other is ICH-GCP, which is the International Conference on Harmonization of Good Clinical Practice. Before we dive into our discussion, I do want to make a note for our United States-based listeners. Polyhexanide solutions, which are what we will be discussing throughout this interview and are what was studied in the publication, are not currently approved by the FDA in the United States for this particular application. The first question that I had for you all really relates to the fact that the irrigation of wounds at the conclusion of a laparotomy is a common practice, and one that many of us have simply accepted as both routine and expected. It's also not something we think of as being terribly costly or time-consuming. So what was it that motivated you to ask the question about its effectiveness in reducing surgical site infections?
Our motivation was that surgical site infections still remain a very relevant problem in abdominal surgery, and that behind many of the practices we call routine or that are routine to us, there's actually no scientific evidence. And we became aware of the fact that the practice of wound irrigation, even among our own colleagues, varies very substantially. And there are some people that rinse the wound routinely with saline after each operation, and there are others that never irrigate the wounds. And there are again others that use antiseptic products like polyhexanide or iodine solutions to rinse the wounds. So we started looking into the official guidelines, and we realized that even in the guidelines from, for example, the CDC or the World Health Organization, there is no consistent recommendations regarding this practice. And this was our motivation to set up a clinical trial to clarify this.
If I may add, there's always some kind of clinical problem you have, and it's something that occurs in everyday's life. You know, you see a lot of surgical site infections and you can't really explain why they're here. And then you start to look around and find strategies to reduce this problem. And this was ahead of the trial. A phase like that was quite difficult, and we had quite a lot of problems of SSIs and having the patients in the hospital for long periods. And then we looked to get along with this problem. And this was also some kind of motivation. And we found the low evidence level and went on to design the trial.
I noted that you all used the existing CHIR-Net trial network as the centers that were engaged with this study. And when I did a little bit of background investigation about CHIR-Net, this is an amazing network that you all already have set up in Germany. Would you please give our readers a brief introduction to CHIR-Net?
Yeah, certainly. So I actually was one of the founding members of the CHIR-Net, and we started all off in 2006, and the motivation was at that time that there was almost no evidence in surgery. And northern countries like Sweden and Denmark and the Nordics, they were far ahead of us in designing surgical trials. And at that time, the output of data and literature from surgical trials was almost non-existent in Germany. And then we were lucky to receive a funding from the German Ministry for Health to establish that network. And in the beginning, there were like 10 university centers, and this developed now until up to 17 regional university centers with satellite hospitals. So we have around 250 cities now and 400 hospitals included in that network. And the aim is certainly to get patients into clinical trials, surgical trials. And so far, the CHIR-Net has randomized around 13,000 patients in 45 trials. And that's quite a success, I would say. And we are happy to have that platform available, and we continue to go on with that.
You all are to be congratulated for that collaboration because it does sound like it's having a really tremendous impact. I try to use these author interviews as an opportunity for junior researchers to learn more about the process of projects like yours. And you all had something particularly interesting occur during your study process. I am assuming that it was an interim analysis that showed an excess of patient dropout due to emergency relaparotomies. And I would like to hear more about how you worked through once you realized you needed to recalculate your sample size and alter the protocol.
Yes, that was unexpected indeed. We had already recruited almost 70% of patients when we realized that the dropout rate was almost twice as high as we anticipated. So we started to investigate what the problem was, and it turned out that the treatment of postoperative complications was very different between the study centers. And there were some that were very pro laparotomy for treatment of postoperative complications. And of course, those centers then lost a lot of primary endpoints for our study. And we didn't anticipate that when we planned the sample size. And then we were, of course, afraid to jeopardize the statistical power of the trial when we lose so many primary endpoint data. And we consulted with our statistical team what to do, and they came up with a new statistical analysis strategy with a new model that included relaparotomy and death as competing risks. And that was a very elegant approach to save the statistical power, and it required to recruit some additional patients, 150 additional patients. So for that, we had to apply for additional funding also. So it was quite a long process, but in the end, it was a good strategy and made sure that we get significant results from the trial.
And I'd like to add that the impulse for the new strategy basically came from the funding body. So the German Research Society and the reviewers said, you cannot go on like this, and you need too many patients to reach the power for the trial. So they actually proposed this statistical strategy with the competing risks.
Competing risks, yes. What was the biggest challenge that you all had in either designing or conducting this study? What was the biggest hurdle that you encountered, and how did you overcome that?
Basically, there were two challenges, and the one in conducting the trial was that the authorities told us that polyhexanide would be a drug. So we had to do basically like similar to US an FDA trial. In Germany, it's called B-Farm. And they told us that we had to go on with ICH GCP conditions and work in compliance with ICGCP regulations. And this was the biggest challenge to, even though everyone was trained but not really experienced in conducting drug trials, to work compliantly with the regulations. And there was a lot of discussions with the participating centers that they had all to record all those adverse events and serious adverse events and reporting them to the authorities and so on. So this was the most challenging thing in the conduct of the trial.
Why did you think that it was important to analyze the bundle outcome that included wound edge protectors and glove exchanges with polyhexamide irrigation?
The implementation of intervention bundles is becoming more and more popular. If you look at the recent publications for surgical site infection prevention, because you never use just one intervention as a surgeon, but you have many steps and they might just add up to the result. And using wound edge protectors has actually been investigated by another CHIR-Net trial a few years back. And it showed that in colorectal procedures, wound edge protectors are efficient. So since then, at our institution at least, we use it routinely. But for glove changing during the operation, there's actually no scientific evidence. As far as I know, there are no trials, and it's more like an intuitive thing that you do. So we were just curious if all these measures combined could maybe have an effect in these types of surgeries, but it turned out that it didn't.
As the investigators, what finding for you did you consider the most interesting?
So we were quite surprised about the result of the trial because we basically thought that there would be a real benefit. To explain the outcome, it's like as it's written in the manuscript, that there were too many clean contaminated procedures. And the main surprise in the trial was basically that the SSI rates were pretty low, around 10% in all groups. And this is a little bit in contrast to a previous single center trial, which was also conducted in Germany. It's called the Recipe trial. And this one showed a clear benefit of polyhexanide in preventing SSIs. However, in that trial, there were procedures more related to colorectal diseases and Crohn's disease with patients who were under immunosuppressive medication, and the contamination grades were a lot higher. So as a consequence, their SSI rates were about 25 to 35%. And this is something which you have to consider in your local practice. So if you have very high SSI rates, no matter what kind of procedure you have, then in that case, polyhexanide might be an option to reduce SSIs. But if your local rates are low or very low, then basically there is no benefit.
My closing question for you is, what do you consider to be your next research priority in the area of surgical site infection reduction?
So there are actually two questions that emerge after the results from this trial. The one is what Daniel just said, to look into the efficacy of polyhexanide irrigation in contaminated or even septic procedures, because we still don't know if the benefit might be bigger. And so we are hoping to do a sequel trial, like a follow-up with contaminated and septic procedures. And the other topic is that the polyhexanide solution can only be used in the wound. It cannot be used in the abdominal cavity because it's not licensed for that. And we had some colleagues that asked us, why do you not irrigate the abdominal cavity? Because maybe the germs that are responsible for the surgical site infection come from inside of the abdominal cavity. So we already started single center trial at our institution looking into intra-abdominal irrigation to prevent surgical site infection with a different antiseptic solution that is licensed for this.
Yeah, so it's important to say that polyhexanide cannot be used within the abdominal cavity. It's not allowed.
That is an important feature. Dr. Mueller and Dr. Reim, thank you both so much for taking time to speak with me today about your study.
Thank you.
Thank you so much for having us.
This episode was produced by Daniel Musisi at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. Thanks for listening.